Hello everyone, welcome back to the Dcademy biology class. In continuation of the previous class, we're going to study the structure of tRNA and translation for today's class. tRNA structure is mamang class ko transcription tawag ko. Mamang class up to this sibok tawag ni the stop codon, initiator codon kana tawag then huna mutation siman to tRNA how to nga sida mutation ni matang sa harim game. Mutation ada di sagi DNA sida may nucleotide change lang to niya. Or deletion will be or deletion will be or addition will be as a sudden inheritable change in the genetic material. Thus, mutation refers to the errors in codon due to change in nucleotide space. Again, nucleotide ke base to change to name. And the various type of mutation. Twin mutation among the frame shift mutation among the twin mutation hata din sagi ko na glutamic acid. Glutamic acid is valid. For example, GUG do. GUG is na kari. GUG is glutamic acid. Glutamic acid siya. Same with the name. Ado mase si single base pair for beta globin change just in the change of amino acid. GUG is kind of valid na one video name. My quote today is complicated. Ado GUG is kay the quote one. Valid ni quote yung siya. Valid niya ate. Valid. GUC will be GUU or GUA will be GUG. GAZ. Okay. I see. Suppose kayo niya glutamic acid siya. Magi di GAZ niya. May quote siya. GAZ o tadi glutamic acid o gada ba siya. Iigi mo ta karisin niya. GUG. GUG. Habis A is replaced by U replaced by single nucleotide change matamda valin haba amino acid yawe. See amino acid all of that D. This is a point mutation. Single nucleotide change tawraga change tawu kai koi khona point mutation na ko. Ado GUG sida GUG on guitar D. This is samana hali sickle cell anemia. Sickle cell anemia hai ta din sagi RBC do sickle shape o sickle shape adi thangon wan de. Ada sickle shape o da de sagi mai numerous changes am na ani ko. The changes single base pairs in the way beta globin change sagi sickle cell o ba adagi hemoglobin reduce torakpa oxygen sorwon bada oxygen namna walakpa blood the oxygen level handarakpa then blood clotting nakpa so multiple characters matang soidarak to nima kamaina soidarak no sai single point nama soibode na to hali single nucleotide ama soibodegi sagi si disease haba si sickle cell anemia se nada ini and a point mutation this is known as point mutation frame shift mutation haba na kai. Such kind of mutations result from insertion or deletions of base pairs that cause a change in the reading frame. Insertion will be deletion will be change of. Insertion or deletion of 3 or its multiple bases will not change the reading frames, but will produce proteins with different set of amino acids or remove one or more amino acids from the polypeptide respectively. So, polypeptides will remove the delete the addition insert and frame shift mutation. Then tRNA. Or tRNA, transfer RNA is no mass translation na chow na loy ko. tRNA structure tRNA is kanana proposed to Francis Crick. Francis Crick haben pasi naman na proposed to Sagi quote ama quote ko mana san haga sagi kano si quote ka na lit tawdo no. Quote adi khani ko codon do AUG GUU to ba quote ka na lit tawdo no. Ada lit tawdo ta quote man lang gani adapter molecule man lang gani naman na proposed to ada quote lit tawdo ba quote ka na no hai matang tRNA na quote so lit tawdo ni or it is known as adapter molecule hani ko adapter molecule ano quote lit tawdo ni. He suggested the presence of an adapter molecule, which function is to read the code on one hand and bind to specific amino acids on the other hand. At the site amadana code Tolin tawni chumna check tawduni si code che aina si amino acid che puraktoine sagi genetic code ki matang da hai ko genetic code na kai haba codon ahum na saiga so for example GUG no kari amino acid code tawre valin na ba amino acid code tawre ado si GUG code che si valin haba code che valin na ba amino acid che pura laktoine ba kadoba mRNA sequence da mRNA gi sequence da mRNA dagi kari sumagodoino mRNA haba pot sidagi protein semagodoino tro mRNA sidagi protein synthesize tawrtoine mRNA si protein semagodoine ada protein is kaino this is the chain of amino acids ki chain ne amino acids mayam na sumaina mane chain of amino acids anan ka doge da amino acid mRNA dagi amino acid sema matang sida jagi for example mRNA the sequence family G U G A U Z adagidi U kana chi mo ma acid lay ya tra da si ahum sina genetic codon odoni habadi GUG oramadi kari amino acid laktoino GUG oramadi jagi valin na amino acid laktoine adagidi AUG oramagana kari laktoino methionine haba amino acid laktoine ana siguma mai code mom lazy ko ada tadi adu tadi adu tadi methionine adu tadi si code sida la iba mRNA dagi protein so matamda resi keno sia mRNA si khangnaba habidi magi code si khangna o sibu chumi brana khangnaba kari laktoino hangda tRNA is a adapter to read the code on one hand and bind to specific amino acid on the other hand ada nakana madana amino acid pura laktoine man amino acid so pura kala ani amino acid ka de pura laktoino hawa da pai bana te amino acid tRNA sida napaga laktoini aduga side amadana amino acid pura la ani aduga side amadana code to mana read tawdoine chumma sina kai mapano phakala sana lai pa the paste phakala chumma yengmudo adain tawdona sigi tRNA sina sigi mRNA gi sequence ya read tawdoine si pa si valin mapada adagidi methionine mapada serine mapada uracil mapada ana mana pura laktoine aduga nakana madana kadoni chummadi kadoni ana serine do thadaramani or valin thadaramani aduga ani do bond form taraga amino acid form tawdoine aduga protein form tawdoine so the tRNA was known as the sRNA tRNA is kind of we soluble RNA and we before the genetic code was postulated genetic code is coming nada kadogi the soluble RNA and code na mi tRNA abadi kano transfer RNA abani ko mai full form D T stands for transfer RNA T stands for CC structure is what tRNA structure sin the structure of tRNA is hana khangas structure of tRNA is the tRNA has a clover leaf structures its three-dimensional structure shows it as a inverted L-shaped molecule the tRNA is composed of these following the tRNA is also structure this is a tRNA magi structure the anticodon loop amma yaure amino acid acceptor n habam yaure T loop habam yaure D loop habam yaure variable loop habam anticodon loop addition na kami side this is a anticodon loop amino acid sequence acceptor sima pasina amino acid sequence acceptor 5' to 3'. D loop this side is known as D loop D loop ada anticodon loop sina kadawi it is a trinucleotide complementary to the other corresponding codon in a messenger rRNA sequence ada sima pasida anticodon loop se yaudune si anticodon loop habadi codon dugi anticodon ai tadi sagi GUG do mana mRNA sigi ina mRNA khalo mRNA की sequence GUG मपासी दा को माना लाख तो इसी ने कहना G की दी मागी complementary base दी कहता है G की दी C U की ना A G की दी C हता सिन अला मागी anticodon से C A C ओरनी anticodon से अला माना चुम्ब्रा ना सी मासी पूरा anticodon याव तो निको कहीं नो मसागी RNA सी की codon मपासी का नप्सन नरुदने खंग नबने और सी मपासी निको सी RNA मपासी को माना खंग नहीं पेस खंग बगी टाइप पेस खंग बगी टाइप तो और सी मपासी यावे दे GUG जिका यावे GUG जिका यावे चिरा तने अरे GUG चिरा तो से करम बक कोडोन करम ब केनो ओरनी न्यूक्लियोटाइड ओरनी CAC this part is known as anticodon ono tene na anticodon ko tanuwa ada amino acid receptor and amino acid mine da ede chena sa amino acid ka nap to ba the amino acid sa pula amino acid na pa ka da amino acid si matoli na pala to ne T loop it helps in binding of the ribosome si T loop sina binding of the ribosome body then D loop it helps in binding of the amino acid synthesis. It is an enzyme. When translation nila ko nai. It is an enzyme amino acid synthesis. D loop ta pulan. Then variable loop it helps in the recognition of amino acid tRNA synthesis. So enzyme is what required in the translation process. Translation process to ore nyawa kani si enzyme ani si. Amana D loop ta lani variable loop. This is a variable loop. Variable loop it is a recognition of the amino acid tRNA synthesis. अरे वो तो सी की उड़ना this is the mRNA tRNA ला है tRNA सी मतोन सी दा कई तपे this is amino acid नत्रा shares is amino acid लगा सी amino acid लगा अरे मागी anticodon ना पासे देंगो माला कोड तो दिशेंगो नसाई ने pH थाकत पदा करम बचुमी ना चेक तो दे दो माना से चेक तो ना मने वाला इसी अरे मागी anticodon सी दी सी खंगदमनिना को आमरा सपोज सी लेते माना पूरा तिसे UCA अदि UCA ना नपतोय मपासे कायदनो UCA ना नपतोय मपासे सगी मागी चानाबा मागी U गिदि कॉम्प्लीमेंट्री होय सिदि U गिदि A लेरे C गिदि काय लेरे C गिदि G लेरे A गिदि U लेरे अदि A G U मपासे कायदनो ना माना लानी अदि सी A G U मपासे सी लेरे A G U मपासे लेरे then bond form tawrani si bond kai oramani hydrogen bond oramani temporary ni ko not permanent hydrogen bond form tawr then hydrogen bond form tawre suna kan site na kari le de ai man tam da serine na ba le serine ya ko adu ngoni si tRNA structure is a tRNA structure anticodon codon tRNA molecules है then translation level processes tRNA याव the name translation level process so translation that is defined as the process of polymerization of amino acids to form a polypeptide chain or can be defined as a process in which the codon present on mRNA codes for protein by guiding the linkage of the specific sequence of the amino acids. Sometimes the protein translation is the process of synthesis of protein from mRNA. mRNA की protein वो translation का. Translation by guiding the linkage of the specific amino acid तो माना पूरा लागा amino का protein सेम भी नहीं tRNA से ना amino acid से पूरा लाके जो tRNA से amino acid से पूरा लागा amino acid chain का form तोड़नी then amino acid chain form तोड़ता है protein वाले then machinery required NKK changi ribosome changi adi tmRNA changi tRNA changi then amino acid tRNA synthesis changi then amino acids adana this sigasa translation tawda ina changba myang then ribosome first ribosome structure the names ribosome is the cell organelle that is responsible for protein synthesis cell organelle that is responsible for protein synthesis it consists of structural RNAs and about 80 different proteins. 80 different proteins. More the structure RNA, protein can more the 80 different proteins here. Enzyme ribosome consists of 2 subunits in its inactive state. Mas in 2 subunit na sai kay subunit ni mo small subunit haban ne large unit haban ne. Large na khar chaw small na pick pale. Small subunit, the function of the small is binding and reading of the mRNA protein during proteostasis. Badika na pala diba mRNA. mRNA ki mapado kaya na khang na ba small unit in mRNA pa. This is a small. This is a small. Si ulo na siya. Small subunit niya. What is the large subunit? Large subunit sina kari puro lam dong malima sina catalyzes the formation of peptide bond between the amino acids. Amino acid spinal amino acid ngam le amino acid amaga amino acid na samdana dun spine. Amino acid amaga amino acid ka samdana iba bond su kai kawi peptide bond. Amino acid amaga amino acid amaga samdana iba bond su peptide bond koi ko peptide bond. Large subunit, this subunit catalyzes the formation of peptide bond. Large subunit na to siya peptide bond form to company. As it provides two sites where amino acids can bind to and be close to each other for the formation of peptide bond. Ada sila to. Si tRNA niya tra. Si tRNA si mana may mapa sila puro yaw lani. Then this is the amino acid. Bond manong niya the amino acid ni. Tugang marak sila. Amino acid ka daw si amino acid ka daw bond form to ini. Sige bond form to siya. Then this is the peptide bond. Jadi kayo ni peptide bond niya. Pagkat si amino acid ka chain mo kayo ikaw na. And so the chain naman form to ni. This is known as polypeptide. Peptide niya na yun ang polypeptide ko more than one polypeptide. Polypeptide later di kayo wala ni protein ulan. Ano ni? Alas dahil this subunit formation of polypeptide bond. Last subunit ito kasi polypeptide bond form to one ba? It provides two sites where amino acids can bind to and be close to each other for the formation of peptide bond. Amino acid yung side bond form to one mapanim. Ribozyme. Next kina ribozyme kasi kayo, no? Ribozyme is the enzyme present in bacteria. Thus the ribosome also acts as a catalyst for the formation of peptide bond. Na kadogeda ribosome masina it acts as a catalyst for the peptide bond. Peptide bond form to mo the mating pa ina. Masuk catalyst na ko dia. Then initiator tRNA. Ini jangan initiator tRNA. tRNA that is required for the process of initiation. Then she will initiate the tRNA code. Adigi tRNA stop code on pura lakwa tRNA related. Translation unit higher kana RNA dagi protein so kada mapadu translation unit. A translation unit presents in the mRNA sequence of RNA that codes for the polypeptide. Polypeptide ki chain wala ka dito. Adugi RNA mapadu kay ko. Translational unit and it is flanked by the start codon AUG and the stop codon AUU and ado ka naging magtalay diba how bugi start codon do AUG na hare AUG malay matonda na ado stop codon na pasi na AUU ado untranslated region ano sagi nangi mRNA kuding magwala translation to kaniya balay tip mRNA ki sequence kaladi translation to the video she mapasa na untranslated region na lakowi. Kaya presented both 5' and adigi before start codon naman dalay bama at saka start codon o stop codon ni matung dalay bama. Stop codon ni matung dadi 3' end. The requirement is crucial for the efficiency of translation process. Series of translation. Translation na kay tawi. Translation na kay tawgida ahamada initiation na rin. Ani shubada elongation sa manay. Then termination. Termination na loiba elongation na chain na form torani initiation na kaya digi hauge do ko. Hamada amino acid chan activators. Amino acid pura lagto ni ko. Kanana tRNA sina amino acid cha pura ga lagto ni. Amino acid pura ga lagpo sida kumain tong gida hamada amino acid activation na ko tRNA pura tan. Formation of peptide bond requires energy. Peptide bond adi kay niba it is the bond between the two amino acids. Two amino acids like bond ko peptide bond ko requires energy ko energy ta kawan. In this step, the amino acids are activated in the presence of ATP. Ada amino acid cha ATP kalau ini agak activate tawani and they get linked to the RNA. Mo coated RNA. tRNA itu mo nakpani. Kaya niba first kada ba T amino acid apa pochi mana side cytoplasm nila lay ni amino acid che. Amino acid che kada ba kada lakto ni ba kada napalay to kido niba. Kada activate tawdo niba. At hagi tRNA sila activate tawdo niba. Fine. tRNA sila napalay ni fine. This is tRNA. And then they get linked to their cognate tRNA. This process is known as charging of tRNA. So kay kawi tRNA get charged to ba na hypo or immunoacylation of tRNA. Immunoacylation ay tadi amino acid hapton ba? Addition of amino acid to tRNA. This is known as immunoacylation. This amino acid bind with the enzyme immunoacyl tRNA synthesis in the presence of ATP and magnesium ions and become activated. Ano amino acid apa po si AA na denote tawre then ATP sina haban ko amino acid activate tawre ATP sina bra ay tadi it uses it required ATP for the activation of the amino acid. Amino acid activate tawre ay tadi ATP data or kada enzyme sida ka or kari enzyme not. Name the enzyme which is used in the which is required in activation of tRNA or activation of tRNA hydroxyurea or activation of amino acid or immunoacylation of tRNA and hydroxy. Immunoacyl tRNA synthesis. And magnesium ions is also required. Ada ani siga pullaga amino acid sa activator ni. Amino acid activator means AMP enzyme complex PPI. Sila. The resulting amino acids AMP enzyme complex is known as the activated amino acid. There is a tRNA. Is there amino acids? Like that. Like there is no amino acids. Then after activation of the amino acid activate tawre. Amino acid activate tawre with the help of the enzyme immunoacyl tRNA synthesis. Then kadorni sige sida magiksi kay kawi carboxyl N ko tra. Carboxyl N do. Carboxyl N sige 3' N of the tRNA, not the 5' N. With the 5' N to the 3' N, 3' N of the tRNA sida masya link to salani bind to salani. Kaya ko ano na? This is known as the activated tRNA or you can say immunoacyl tRNA synthesis. Immunoacyl tRNA synthesis. This process is known as charging of tRNA. No na ako na amino acid or tRNA se activate tawre ko. Ano ba translation thokri? Translation na matamda process to step kya laiba initiation, elongation, termination. Then first initiation initiation nito tawre si kayo. Large and small ribosome subunits mRNA initiated tRNA and 3 initiation factors IFs are required in prokaryotes to start the process of translation. Translation si tawna ba kay changye ribosome mo changye tRNA mo changye mRNA mo changye initiated tRNA changye are 3 initiation factors that is the IFSC mari po siya changye. The initiation process are basically 2 steps. Kasi recognition of the start codon AUG. Start codon se kaya diyaron din no mRNA diyaron din ko. Start codon se kaya iniba sa AUG mapasa chiba la ani. Panana tRNA sina. Ano tadi anticodon di kayo rin. tRNA sila anticodon yaw din na tRNA sida sida anticodon yaw laktune adi start codon sida AUG ore AUG otadi magi anticodon di kai orani A gi complementary base di A gi complementary base di U lere U gi di A lere Z gi di C lere ada U is C ne ada what is the anticodon of the start codon hai tadi anticodon of the start codon is UAC ada tRNA sini kadoni si UAC sa UAC yabu tRNA sa pura lani kada start codon of the mRNA ki start codon sida pura lani then binding of the ribosome and mRNA and the start codon binding of the ribosomes and mRNA at the start codon adadi start codon no pasi de osile start codon pasi kanana pubiraktoine ba large subunit of the ribosome gi large subunit na puraga laani siya pasi leire ada before that sigi large subunit sida akoina kadoge the site ahung thokna khaide E B A side ba ne P side ba ne this is exit side this is E side exit tawde ada AUG na first P side sida sino pa sida P side le sino pa P side na kadoni sida tRNA to la lakto si kalam bus side to kai ne ribosome je kone side hum tolong kan de E side ba ne P side ba ne A side ba ne ada tRNA na lakto si tRNA yaw to si kai de no jagi P side ki ma pas de ne ada kalam P side lakto ri ba tRNA si kadi code pura ga lakto ni ba UAC ha ba code sia pura ga lakto ni ada UAC de si le then AUG na pas de then UAC da pura lakto si kai de methionine na sai kai hagi AUG haba kano si codon sina kari amino acid na mana code to de sia amino acid methionine ne duna dan sila di anticodon je ngono sigi mRNA gi kano si ngo codon je ngo mRNA gi AUG na pada lang so si methionine na amino acid pura na ne by the tRNA having anticodon UAC this is mRNA ko masi codon hung pona khaite ko chain du pulong polypeptide chain lebido sida long chain of RNA na tra se si chain le se polypeptide chain ja si china le de man polypeptide chain hai de steps involving chain initiation are a complex is formed by the attachment of small ribosomal subunit to 5' end of the mRNA. So start codon do 3' end romdra 5' end romdra le se start codon le se 5' end romdra le not 3' end nate. At the 5' end of the mRNA, the start codon AUG lies downstream to this mRNA, which initiates the translation process. AUG se kai kana ki downstream da leiba si mRNA ki downstream da si pulap si mRNA si downstream da kai le ni AUG ha chile joining of the initiator tRNA with the start codon by its anticodon through hydrogen bond so the anticodon si pura la UAC sina adu UAC siga AUG ga bond form to this what type of bond hydrogen bond formation of translation initiation complex by joining of last ribosomal subunit with a small ribosomal subunit. The ribosomal amadri samnaraga leiba natteba sidabudi samnare. Samnaraga leiba natteba ko. Sini. Ada kadoge da samnaraga leiram da basa. Atagi initiation how tardi once the RNA sina AUG mapada bond form tawraba aduga small subunit laani laga small subunit laaga kadoni polypeptide chain formation je hawadi formation of the translation initiation complex by joining of large ribosomal subunit with the small ribosomal subunit at this stage two sites are defined. And the P site A site P site the site of the ribosome where initiated tRNA lies. That is the immunosal tRNA site. The immunosal tRNA is having the tRNA is called tRNA no immuno acid proper tRNA the name. Aduna immunosal tRNA. That is activated tRNA. Activated tRNA से पूरा गला आनी activated tRNA से पूरा गला आनी and A site में पास ना the peptidal tRNA site which lies vacant for another charged tRNA to enter the C में पास ना another tRNA dong lock from the power in nearby A site अगर सी E site ना कदोगे तो C से methionine thanamani ko sige tRNA se methionine thamamani sige next tRNA na malani aju code form tawgani aduga manasu sige amino acid am puraga lani activated tRNA adu sige methionine ani si bond form tawgani bond form tawga sige pocha exit site ta kidune so matang la tRNA lani sakani tRNA lani etc right then elongation the tosh elongation is kaya no during elongation of a polypeptide chain, the amino acid tRNA complex sequentially bind to the A side of the ribosome in which the appropriate codon in mRNA bonds with the tRNA anticodon by complementary base pairing. Aduna kaidore nga sagido sida methionine sila ko another tRNA activated tRNA or immunosal tRNA is brought to the A site of the large subunit of the ribosome की last subunit यावबा A site sida lani laga kari form ta bond form sida to kari ma for example si ACA ko CA my anticodon di kairani U sida ACA ore ACA ki anticodon sidi UGA ore UGA ore ba anticodon pura lani ACA sida kari enzyme kari amino acid ko threonine have amino acid se pura gala ko si amino acid nappe then with the help of the enzyme peptidal transferase with the help of the enzyme peptidal transferase a peptide bond is formed between this amino acid the methionine with the threonine threonine ga methionine ki marakta peptide bond form tawgani peptide bond form tawre by enzyme with the help of the enzyme peptidal transferase between the carboxyl group of the amino acid present at P site and amino group of the amino acid at A site. Sige carboxyl group of the amino acid carboxyl group of the amino group of the sina amino group ko yi sina carboxyl group or carboxyl group ada kadoge da mana hai se amino acid ko mana carboxyl group di P site la riba carboxyl group sida di P site la riba carboxyl N aduga kaidoba A site la riba amino N ani do bond form tawgani that is peptide bond form tawgani. Addition of amino acids takes place one by one in the sequence of codons and thus translated into the polypeptide sequence that is dictated by the DNA and represented by the mRNA. Adaman hai si kaidawani sidagi after that si once bond form tawgadi sigi tRNA si exit site tat khani dugo ma remove tawgani another adugo sigi potcha sigi kadawani si sumana khumjale nang khalo atare sigi P the methionine le rambasya P the methionine le rambase kadoge da sida laani sigi potcha ti methionine thanamani thana si bond form tawge tRNA se si ma pa se laani sigi tRNA se sona kala ka si next le riba tRNA se P site ta laani P site ta laani ko sigi kenu sia suppose sigi tRNA se si E P E na lai ko ada ahambagi AUG pura laamba CAUC oiba anticodon si exit site laani adagi sida methionine da malik sida bond form ta hore bond kana bond form ta hore threonine ga bond form ta hore ada threonine sida napa DNA ka dong laani ba A site ta mu P site ta mu laani then C T site la another DNA yung laani another DNA yung laani sida kai atokpa kainomang pura laas imino sidang pura laani ado si kai dong laani A site dong laani A site dong laani ko then si A site lakta di sigi potcha si remove tawani si mangani and sigi potcha sidang laani laani aduga 시제 가더니 시다 자기 마이 보다 타나머니 에다 미ಥionine laire ida threonine laire ida another sida porokpa keno sia immunology support sina keno haisi histidine haisi histidine na sida hang thawa hari ko histidine yaw pochi ka da la ani ba A site da la ani dana sain to mathang transfer to na ga swain polypeptide chain P site da la ani exit site da kani P site da la ani exit site da kani P side da laani exit side mathang ahambada A da laani P da exit sakani A da laani P da exit sakani like senior and then the ribosome more strong codon to codon has when the ribosome more strong codon to codon along the mRNA in 5' to 3' direction kari direction is very important kari direction is 5' to 3' direction additional amino acids takes place one by one in the sequence of codon and thus translated into polypeptide sequence, which is dictated by the DNA. DNA na, asay dictated by the DNA abdi ko no si mRNA si kada siya mo kigi. mRNA si with the transcription ni matong inna, asay DNA digi RNA siya mo kiba na tro mRNA siya na. DNA digi mRNA siya ni ba ni mRNA digi protein siya mo paneba. Ado na DNA na dictate tawraga kay ole mRNA na represent tawraga amino acid chain form tawre amino acid chain form tawadi kai ola do na sa si kai ola ni protein ola ni chan to na sigi sumcha sisi protein mana sumcha kidonya sumcha khaga until kadalo de ba UAA haba stop codon ji yaura ganda di translation sa loshalani loani das in gani PA da sai transfer ti ai sum among la ga chakani among la chakani then termination number sina translation complete order the process of translation or protein synthesis terminates when the A side of ribosomes encounters a termination codon as a result of which no DNA further binds to the A side of the ribosome. Ada ka daw ni, masagi stop codon yaw tadi, stop codon sila na. Stop codon ni yaw na ba? Ado tadi A side ta niya. TRNA ang talaro. Ano ba TRNA na kasi kaya no? Sige A side sila TRNA sila lakto niya ba? Ado mo, stop codon sana sige ribosome sina zara to matam sida mRNA di karamba site. Stop codon yung pasi yaw tadi. Stop codon yung pasi yaw tadi sige si kari ba? A side sida TRNA talaro. A side TRNA talaro. In which no DNA further binds to A side of the ribosome. Nice. Finally, a release effector binds to the stop codon and translation is terminated, thus releases the complete polypeptide from the ribosome. So, release factor then proline the finish story. Proline the finish story and the CPRA released this is a protein. So, na karoon di ko machat leba 5' N to 3' N. This is the ncert textbook to yawo. So the exit site mo na di si haptre. Then kay haba si exit site. is P site. A site. Sim. Ada pura si cakap selalu cakap dia ni kok. Si alanan siya. Ada transporter ni paling tak tu peptide form tahu ni. Once si buon form tahu kita di TI ni serang cakap ni. Dan serang cakap kita di si TI ni sondong lah ni. Si TI ni sondong lah. Macam transporter ni. So transcription is finished. Translation is finished. Translation chỉ rồi đấy. Rồi là cái không cái tàu grab trong division chỉ translation chỉ cái đấy however tRNA trong đó đi activation of amino acid đó amino cell tRNA synthesis đó cái tRNA sẽ activate đó ni amino acid pura cái là ni then adu amino acid itu cái amino cell tRNA nó kau dah adu cái initiation da sa sky degi hawduna start codon ji kaina AUG then elongation process tawni elongation taw se sai ribosome ko MR last samena du ribosome matung inna kon angang da protein ji ka da synthesize taw hagi protein na synthesize di ribosome hagi other ribosome jina with the help of the ribosome jina ribosome la ta di tRNA sula tRNA la ta di tRNA lak ta di protein sungare ada ribosome si important role ne mRNA de ka do ni ribosome na sala la ni na sala ga bagi E side B side A side na ko last one de khai do ga pedo da B side da first B side da like du lak to share tRNA with the as in complement having anticodon ko UAG ka anticodon oi ba pochi pura la ni UAG ka anticodon le badi kai ne ba UAC yaw ba or methionine na pagkalaan is methionine na pagkalaan. Methionine na pagkalaan then amino A site mo next amino acid mo lani ang sumayin tayo na sa chain form to ni next site amino acid lani then elongation form to ka until sa stop ko don UAA haba poche yaw ka stop ko don sil loira ka terminate translation check finish well. Translation is completed. Okay, up to this mathangi class da gene expression ji tawsi. Stay tuned. Have a great day. Bye.